So now we're going to go on to genetics. We'll talk about Mendelian genetics, the way that any trait, including but not limited to a disease, would be passed down from generation to generation. And depending on the way that disease spreads, is it going to be dominant? Is it going to be recessive? What are the statistical chances of offspring developing that gene? We're also going to talk about the actual DNA and mutations and possible repair and what those mutations are going to look like. So we'll have a little bit more time for principles of inheritance in a bit. So we'll do that Pinet Square a little bit later. Raise your hand if you have never done a Pinet Square. One person's never done a Pinet Square. Anybody else? Everybody's done a Pinet Square? Excellent. It is a learning objective. You need to be able to do those. So let's just go quickly back to this concept of the structure and function of DNA. We've got our double helix or strand. What's that? What's that face mean? There's a Kendrick Lamar song about it. Okay, DNA double helix. That means we've got two strands of nucleotides. They're actually going in opposite directions with base pairs towards the middle, and they're going to match up those base pairs so that if you have one half of the strand, you can always write the other strand, right? If you have one half of the DNA double helix, you always know exactly what the other half of the DNA double helix was. That's really important when we undergo mitosis and replicate DNA. I'm using some high-level terminology here because you should already own all of that terminology. Are we all happy and comfortable with the way I'm describing all of this? More or less? Okay. So a DNA mutation means that one of the nucleotides has been damaged. Yes? Oh, sorry. I think I moved it.
It's in slide 105.
Yeah, and it's probably 105. It's probably better off where it is later on, actually. I think I changed that later to put it in a better spot, and probably changed it on campus. Or it's in a better spot here. We'll figure it out which one it is later. I'll tell you later which one it was. So DNA mutation and repair. Your body may be able to repair damage. Now, again, that DNA damage came from ionizing radiation or toxic exposure, anything that can physically knock it out of the way. Viruses can knock it out of the way. Lots of things can do that. Your DNA polymerase may be able to heal that. It may be able to replace the damaged nucleotide appropriately, or it might not. So here's some common ways that genes mutate. Now remember, every 3 nucleotides, we're going to call that a codon, because every 3 nucleotides codes for 1 amino acid. This guy gets it. I don't know your name yet, but you look like you know what you're talking about. Okay, so you've got 3 base pairs. That represents 1 amino acid in your final protein that you're going to have later, right? And we've got a genetic code. We've coded it like a computer program. We've figured out every 3 base pairs and which amino acid corresponds to that. Which means we've figured out if we change a single nucleotide in a sequence, what it's going to do. And depending on which mutation occurs, it's going to be something different. So let's see, Arfilupro. Okay, so the first thing that could happen, or not the first, but one option is you can have a silent mutation. Some of these codons, some of these sets of 3, code for the exact same amino acid. It's the luck of the draw that sometimes a mutation occurs. We replace an A with a G when we repair this DNA sequence. Whoops, that was a mistake, but it's silent because you get the same exact amino acid out of it. Silent mutation is a possibility. And then you reproduce, your kids get that gene, they make the same protein. The IKEA chair turns out exactly the same way as an IKEA chair should. Or you got a slightly different screw. Yeah, it works. Chair works. What's that? It's not detrimental, correct. The next 2 are both considered single point mutations. In both of these next cases, only one nucleotide changed, but the outcome is very different between a missense mutation and a nonsense mutation. A missense mutation means instead of getting this amino acid, we get one different amino acid. Sometimes one amino acid is not enough to make a non-functional chair, but sometimes one wrong amino acid is plenty enough to make you have non-functional chairs for life. So sickle cell anemia is the product of a single point mutation. So this is one case where protein doesn't work anymore. Hemoglobin is the protein that we're talking about in this case, right? A single point mutation is enough to completely mess up this person's life. In this case, that won't always be the case. There are some genetic disorders that are pretty mild in comparison. The other single point mutation is a nonsense mutation. Like I said earlier, this is a code. We've coded it like a computer program. Just like a computer program needs a place to start and a place to stop, if you don't have a stop point, that code just keeps going forever, right? It's a computer, it doesn't know to stop. Just like that, your cells are a program that doesn't know when to stop unless you tell it to. So there is a sequence of nucleotides that represents stop. Just like a computer program will have a stop condition, an exit condition, right? So in this case, just by chance, luck of the draw, this mutation happened to give you the stop codon, and now instead of building the whole chair, you built half a chair. You just stopped too soon. So still non-functional chair. So hemophilia for factor 7 would be an example of a nonsense mutation where just a single point mutation means that this person can't clot properly. Okay. Yeah, so you would have had a stop sign somewhere over here. Instead, the stop sign's over here. There are start codons as well. Okay, so frame shift mutation. Now, in all of these cases, we took what was supposed to be a TTA and we changed one of the base pairs out of that, right? But we changed it to something else. Something did get replaced. DNA polymerase came in and it put a nucleotide in place, but maybe it put the wrong one in these two cases, right? Or put the wrong one in. What if that did not happen? What if DNA polymerase did not come in and repair that, and instead of reading, a T and an A, or a T and a T and a G, or a T and a C and an A, you frame shifted? And what I mean by that is we skipped pink from here to here. We went to T to pink, A to C to C to T. Now all of those sets of 3. Did that make sense? Nope. Okay.
I can't see your point.
Oh, you can't see my cursor. Sorry, guys.
I'll get my
Sorry, guys. I'll get my highlighter up in just a second. So this is a frame shift. Here's the T, 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 T. Oh, that's all good, right? It's supposed to be a T, it's still a T. This one got changed to a C, but then there's an A, right? What this is showing you is we just skipped. Oh. T, A, C, C, T, A, C, C. Now instead of having this next one be CTA, or sorry, this next column is not going to be CCT. Oh. Next one's a CTA. Yeah. It's going to affect every codon after. Yeah. Now every amino acid after that mutation. Let me get my cursor. So this frame shift mutation means every single codon, and therefore every single every single amino acid after the one mutation is going to be altered. So you don't even have a chair anymore. You've got like a Volkswagen Beetle. Like, it's totally wrong. Or just a pile of sludge, probably, is the more likely scenario there. It's not going to look like an IKEA chair. Just say that DNA polymerase healing did not work in that occasion, and I don't know why DNA polymerase would not work. There could be a variety of reasons for that. Maybe there's a different cell injury. Maybe it's an enzymatic problem. Maybe there's a problem with the genes decoding for DNA polymerase. Like, it's all inside. It's all dependent, right? So radiation, chemicals, viruses are all potential mutagens that could lead to one of these outcomes. And it's luck of the draw which one you get. These can happen spontaneously in an individual. Some genetic disorders have no family lineage, even though it's genetic, and now that person can now pass it on to their offspring. Let's do some Gregor Mendel, Mendelian genetics. So there's some vocab here that I do want you to understand. So doing a Pinet Square has significance in terms of understanding how diseases spread in families, and we will cover that. Part of it is just understanding the vocab of phenotype versus genotype versus, we're going to talk about karyotype in a minute, and then again how that spreads. So phenotype is about outward experience, what gets expressed. You know you have two sets of DNA. You got one from mom, you got one from dad. And for many of these traits that you express, it's the dominant trait that was expressed. Or maybe you have two recessive genes and you have the recessive phenotype. So blue eyes is a recessive phenotype, right? So I have blue eyes, therefore I am showing the blue eye phenotype. That means my genotype is that I have two copies of blue eyes in my genome. Right? But these are not synonymous, right? You can have a phenotype for brown eyes, but you may be carrying the genes for both brown eyes and blue eyes. Your genotype, in that case, does anybody know the vocab word for when you have one dominant and one recessive gene? Heterozygous. Well done. So the genotype is the result of the actual genes that you have, and the phenotype is what actually is visible, expressed, not always visible, because we talk about eye color for these things, but we're also talking about chloride ions. We're talking about all of your receptors, all of your proteins, all of your enzymes, all of the functioning parts of your cells. So did you get your dad's chloride ion channels, or did you get your mom's chloride ion channels? That's not something we always pop into a Pinet Square, like on the first day of Gen Bio, but it's something we can actually pop into a Pinet Square. Let's see. So remember meiosis from back in the day? Yep. So meiosis, we're going to produce daughter cells that actually are genetically different from what your mom and dad gave you, right? It's a remix of what your mom and dad gave you. It involves independent assortment of the DNA that your mom gave you and the DNA that your dad gave you. Sound familiar? Okay. That's what gives us that Pinet Square. So we're going to go back for just a second to this idea of the Pinet Square. It is tied in with the idea of meiosis and how we generate germ cells. So what this is saying is your mom gave you one gene and your dad gave you one gene. And those are your genes now. And it's 50-50 which one you pass down to your offspring through the process of meiosis. While you're undergoing your 2 rounds of meiosis, you are independently assorting your paternal line and your maternal line of DNA. So it's 50-50 whether or not you're going to give that gene to that germ cell, to that egg, or to that sperm. That's why the Pinet Square looks the way it does. Because so here's your genes, here's your partner's genes. You have a side from your mom, you have a side from your dad, they have a side from their mom, they have a side from their dad, and it's 50-50 which one made it into the germ cell. Good? Yeah. So the way we set up a Pinet Square is we put our the genes we have on one axis, the genes our partner has on the other axis. Let's say it's about blue eyes or brown eyes. I've got blue eyes. I have recessive genes. So my axis looks like this. Here's me, little b for blue eyes. We usually put the recessive in lowercase. I largely completely ignore formatting for Mendelian genetics. I'm not a geneticist, it's not my thing. But for this one, it's easy enough and I'll do it properly. Let's say my partner had brown eyes. Now there's 2 ways to have brown eyes. You can have Both genes came from people with brown eyes, so it's it'd be big B. You got two copies of brown eye genes. Either way, that sperm cell breaks down, he's giving me the brown eye gene, right? So how does this play out if this were the case? Tell me if you need me to make this bigger or higher up. What's up? Nope. Yeah. So I always do recommend that you write out an entire Pinet square if I ask you a question based on a Pinet square. Even if it's intuitively making sense to your head, sometimes it plays out differently. So even though this is super obvious to those of us who've done Pinet squares a bunch of times, we're still going to fill out every square. So what goes in the square? Big B, little b. What goes in this square? Big B, little b. What goes in this square? Big B, little b. What goes in this square? Big B, little b. Okay. 100% chance of having brown eyes. The phenotype is always going to be brown eyes. What's the genotype? What's the vocab word for genotype of that kid? Heterozygous. What was my genotype? Homozygous recessive. Excellent. And what was my partner's genotype? Homozygous dominant. Excellent. Good clarifying for yourselves. Okay, let's say my partner had brown eyes, but they carried the recessive gene for blue eyes. Okay. What goes here? Big B, little b. Big B, little b. Little b, little b. Little b, little b. How many of my kids? 50-50 brown eye, blue eye. Yep. So if I asked you how many kids have blue eyes, you would say 50%. Half. Yeah. Obviously, this doesn't work with every single thing. If I were to say green eyes versus blue eyes, it's not always this obvious with recessive and dominant. There's such a thing as codominance. There's such a thing as incomplete dominance. We're just not going to mess with any of this because this isn't a genetics class. What's up? Milkman. No, just kidding. Luck of the draw. Sorry. Sorry, that was really inappropriate. But honestly, if we do sociological studies, it happens more frequently than we would like to admit. There is that possibility. If you look like your parents, don't worry about it. That's come up before, though. We've done like this for like weird recessive traits, like having attached earlobes instead of separated earlobes, and somebody's been like, weird, this doesn't match my parents at all. And I've been like, hey. Oh, okay. Well, anyway. Yeah, so yes, you're expected to be able to utilize a Pinet square to determine how likely it is to spread a disease. Give me that percentage accurately. I think it's on your worksheet today, if I recall correctly. It's a new worksheet, so I may not be remembering. Okay, so that vocabulary, genotype versus phenotype, the homozygous recessive, homozygous dominant, heterozygous. Is there such a thing as heterozygous dominant or heterozygous recessive? No, because you have one copy of each. So hetero just means different from, homo just means same as. That's what those root words mean. Okay. So you're going to have this like sort of pedigree idea of how different types of diseases are going to spread through families. Some genetic disorders are going to be dominant and some genetic disorders are going to be recessive. If a genetic disorder happens to be a dominant trait, what we see in pedigrees is that they do tend to show up in every generation. Typically, it is possible to have children who don't have that, right? If you have the recessive form of the gene that is the healthy version, and your partner also has the recessive form of that gene that is the healthy person, then again, there's a good chance that some of your offspring will not express the disease. But any child who has a copy of the disease will then express it, right? So that one's pretty intuitive. Don't learn these charts. There's no reason to. It's just the general concept of an autosomal dominant pedigree means more likely to pass on to children. Autosomal recessive pedigree charts, These are the ones that typically skip a generation, using air quotes here. You may have heard, oh, this disease skips a generation. It's the one that can lurk in the genetic code, and as long as somebody's got the dominant form, the healthy form of the gene in the family, it's unlikely to show up, but it can if enough people have the recessive form. There's something wacky going on with this chart. What is it? So here's generation 1, here's generation 2, here's generation 3, generation 4. They got with their cousin. That's exactly what happened. And that is to the point because genetic disorders do tend to stay in populations. So populations that are very insular, let's say Hasidic Jewish population of New York City, they like to stay within the Hasidic Jewish population for marriage. And that makes for a very narrow gene pool, and that makes an increased probability of recessive pedigrees coming into play. And it's not just these, and I'm not trying to pick on this one population, right? It happens all over the place. Any sort of ethnicity that's very insulated is going to have this impact. And this happens constantly. If you own a purebred dog, the pedigree looks a lot worse than this, actually. This is what dog pedigrees look like. I've got a German Shepherd and a Black Lab. You better believe they are inbred. And that does mean that, you know, a dog that's purebred is going to be much more prone to having a number of recessive disorders that express themselves. Purebred dogs are just a terrible plan. Yes? There is a genetic basis for twins, but it's poorly understood. I don't know that we've tracked down exactly what that genetic basis is. And I don't know if I would tag it to one like recessive dominant thing. I don't know. You could, probably. I don't know enough about it. Yes? No question, just twiddling things? Yes.
So basically for me, you just wanted us to take that autosomal dominant is more likely to be in every generation, and then recessive is more likely to skip one?
Yeah, I mean, there's that concept. If you can do a Pinet Square, then you can figure this out for yourself.
Okay.
So the concepts tie together. That's all we're going for, is make sure you understand it.
Okay.
Yeah, that's the easiest way to say it is skips a generation, doesn't skip a generation. Now, these are all autosomes. What's the difference? What's the other option aside from an autosome? How many autosomes do you have? You got 23 chromosomes in a pair from each parent. 46 total, right? But how many of those chromosomes are autosomes? That was great. No, out of 23, how many are autosomes?
Well,
All of them.
22.
22 is the correct number.
Yes.
You
The
got
last one is the
sex chromosome. Yeah.
I was like, I know this.
I like the really random numbers you get shared.
Number 3, my lord.
72. No. Okay.
Sorry.
It's okay. It's 4:15. Any laughter is good laughter. So you have 22 autosomes. The last one is the sex chromosome. I think it's kind of funny that we just decided anything that's not a sex chromosome, we're just going to call an autosome, as though sex chromosome is like the standard, and then everything is held to that standard. It's kind of weird naming terminology and definitions there, but that's how it worked out. So all of these would mean it's chromosome 1, or it's chromosome 2, or it's chromosome 3. That's the location for each of these genetic disorders is on one of the 22 autosomes. Our last option is for it to be on a sex chromosome. So fun fact, X and Y, they don't look like an X and a Y. The difference between an X and a Y chromosome is that the Y chromosome is really, really short. So men are just women who aren't really enough, is what we're getting at there. Yes, Fern. You're just less. That's it. That's all there is to that. So when we say sex chromosome linked genes, it's almost always going to be on the X chromosome just because there's physically more of it. So after I burn men, I'm going to burn women. It's our fault, right? It's the sex-linked recessive and dominant degree disorders. They're almost always on the X chromosome. And I actually don't care about X-linked recessive versus X-linked dominant. I just care about X-linked. That's the only thing that's really going to come up. One thing we find with all X-linked disorders is If you've got a second X chromosome, which is to say if you're phenotypically female, then that second X chromosome tends to be protective for whatever this disease is. So as an example, color blindness tends to be an X-linked disorder. And color blindness tends to impact men a lot more than women, because women will probably have two X chromosomes and women will probably have a correct version of that DNA. Whereas men, they just have the little dinky Y chromosome and they only have the one X chromosome. So if they've got a messed up X chromosome, they don't have the other information, right? So two X chromosomes means, well, this IKEA instruction booklet is messed up. I'm going to use this IKEA instruction booklet instead that's not messed up. We got a backup plan, exactly. Whereas, you know, the person with the XY, they've just got the messed up instruction booklet and nothing over here. So that's why it tends to impact men more than women, even though it's X-linked and you want to associate it with women.
Because of the protective factor.
Yeah, it's the other X chromosome is considered protective.
So then the dad gets it, or so then the man gets it, but then he doesn't pass it.
Right, does he have sons or does he have daughters then? Because if he is copulating, reproducing with somebody with a woman with healthy genes, then the daughters will have a copy of the healthy gene. But the sons, yeah. It depends on how that goes. Okay. Is that all making sense? How those things go in families? And again, I want to emphasize, yes, genetic diseases will be carried forward, but a genetic disorder can occur spontaneously in just one generation. Marfan syndrome, a large percent of the time came from nowhere. No family members had Marfan's, and then suddenly somebody does. There's a percentage, but I've forgotten it, of how often that happens for Marfan's. Okay, that's not the only way to have genetic issues. Back to meiosis. This is probably why I rearranged some of these slides, by the way. Again, meiosis is about independent assortment. You have two rounds of meiosis. Round 1, round 2. Meiosis 1, meiosis 2. And actually, let's go back to reproductive lecture, one of the best lectures of AMP2. Almost break time. I'll finish up on this one and then we'll go on break. So remember AMP2, we talked about meiosis in women and how for women it's not at all what you were taught about meiosis, right? For time frame, for outcome, for the number of germ cells, it's not that. What's weird about meiosis in women? What's happening during childhood for the time frame of meiosis? It's arrested. So we've got like our primary oocytes before we're even born, but they are arrested in meiosis 1 throughout puberty. You're there for a really long time. And same thing with meiosis 2. It's arrested until fertilization occurs, and only happens if fertilization occurs, right? So you have these long periods of time of arrestation if you're genetically female, if you're producing gametes and all of this. So a non-disjunction disorder is another way that we can have a genetic problem, and this is going to be chromosome-wide. This is going to be a problem with the number of chromosomes. So non-disjunction means that while we were separating, when we were undergoing meiosis, and we were in the part of meiosis where we separate the analogous chromosomes, that failed to happen. That event is called disjunction. Disjunction failed. You had non-disjunction. And that means instead of having one oocyte with one copy of this chromosome and another with the other copy of this chromosome, you ended up with one oocyte with 2 copies of this chromosome and one oocyte with 0 copies of this particular chromosome. Now again, you've got 46 total. So this is happening individually with any one of the chromosomes, if that statement made sense. This is just representative of this is happening to one set of chromosomes. So this is a non-disjunction event in meiosis 1. You could also have a non-disjunction event in meiosis 2, which is represented here. We separate the sister chromatids, and now we've got 2 copies on this gamete and no copies on this gamete. Now I brought up women first because we're arrested in meiosis 1 for a really long time. That means we accumulate toxic exposure throughout a lifetime, radiation throughout a lifetime. There's a lot of time for this stuff to accumulate. That's not to say that non-disjunction events are only in women. It just means we have the time to accumulate those events. But if a man happens to like irradiate his testicles and then reproduce that night, then it's just as likely that he'll have the non-disjunction event occur, or toxic exposure, or whatever it is, because men are continuously producing sperm throughout a lifetime. The meiosis and mitosis is continuously happening. So women like kept that lottery ticket around for 20 years and messed it up, but men are constantly playing the lottery over and over and over again. So it can happen in either gamete. Not going to put the blame one way or the other. Okay, I'm going to stop for break now. It's 11:52.